As I got older, I came to know that chemotherapy was a very common cancer treatment. I knew it was an aggressive treatment with many debilitating side effects, but I never thought about how it worked or what the goal of the treatment was. After volunteering in the infusion center of the Wilmot Cancer Center throughout the last year, I started to wonder about the science behind it. What did the drugs actually do to the patients' bodies? What was the point of the treatment, and why did it have these effects on those who received it? For this project, I chose to study one specific drug used in chemotherapy. I came to learn that chemotherapy takes advantage of the intercalating abilities of certain ligands. Intercalation is the insertion of molecules between the planar bases of DNA. Basically, the ligand inserts itself in between adjacent base pairs. For the intercalator to fit, the DNA needs to unwind a little bit to open a space between the base pairs. This unwinding causes the base pairs to separate or rise, creating an opening and lengthening the DNA strand a little bit. The degree of unwinding or change in length varies depending on the ligand being used. The intercalator induces structural change that leads to functional modifications, which often leads to the inhibition of replication. The inhibition of replication stems from the interactions between the intercalator, the DNA, and the enzymes involved in DNA replication, such as topoisomerases and RNA polymerase. Topoisomerase's job in DNA replication is to break the DNA to relax supercoils or untangle strands and then reconnect them in the proper form. Intercalating agents often block the reconnecting step, which prevents the successful production of mRNA, which in turn inhibits DNA replication. In this case, a double-strand break is left in the DNA, and this is lethal when the cell tries to go through replication. One specific intercalator used in chemotherapy is actinomycin, a cyclic modified peptide. Actinomycin is derived from the bacteria Streptomyces and was first isolated by Selman Waksman and his co-worker H. Boyd Woodruff in 1940. Dr. Waksman won the Nobel Prize for Physiology or Medicine for his discovery of streptomycin. Dr. Waksman first discovered in 1923 a soil bacteria that was killing other common bacteria growing around it. He didn't begin further investigation until 1937. Waksman and his team studied the bacteria's interaction with E. coli. They found that eventually, the bacteria was able to inhibit the growth of E. coli. The antibiotics were extracted from the culture, and Dr. Waksman named it actinomycin. Originally, the team thought actinomycin was the most active antibiotic ever discovered, but it soon proved to be one of the most toxic ones. As it turns out, actinomycin was too toxic for general use. It effectively killed cancer cells, but poisoned the patient at the same time. Related molecules, like actinomycin D, are, however, widely used for chemotherapy. Actinomycin D was approved by the United States Food and Drug Administration in 1964 and has been used here as a treatment ever since. It is on the World Health Organization's list of essential medicines, a list of the most effective and safe medicines needed in a health system. Actinomycin was the first natural antibiotic isolated that proved to have anti-cancer activity. Actinomycin D is made up of two components: a flat phenoxazine ring that resembles DNA bases, and two cyclic polypeptides that are composed of unusual amino acids. The ring is able to fit in between bases in the DNA helix, and the cyclic polypeptides fit perfectly into the minor groove of the DNA. The interactions of this binding produce a stable, lethal complex in the cell system. The flat ring inserts itself between base pairs and is able to make two hydrogen bonds with guanine base pairs on each cyclic peptide. This favorable interaction between actinomycin and the base guanine causes the drug to preferentially intercalate into guanine-cytosine-rich regions. The interactions also stabilize the topoisomerase DNA complex, which prevents RNA polymerase progression. Conveniently, many GC-rich sequences are found in transcription promotion regions. By intercalating here, actinomycin effectively suppresses the production of mRNA and consequently inhibits DNA replication. The structural changes induced by the insertion of actinomycin D are shown here. Normally, in DNA, the rise per base pair is about 0.33 nanometers. As you can see here, with the intercalator present, the same two base pairs are now approximately 0.66 nanometers apart. The length of DNA strand overall has increased by around 0.33 nanometers. This slight structural change disrupts the processes of enzymes involved in replication. Research has shown that actinomycin physically blocks the enzymes and also may generate double-strand breaks. Although the peptide has anti-cancer activity, it also has toxic effects on humans. Many of the observed side effects of chemotherapy are due to the toxicity of actinomycin. Actinomycin can also be used for research purposes. A related molecule, 7-aminoactinomycin D, is a fluorescent derivative of actinomycin. It is able to be used as stains in microscopy experiments. The molecule stains GC-rich regions and binds to single-strand DNA, so it is useful in determining apoptosis and distinguishing dead cells from live ones. Actinomycin has been used for a very long time and will continue to be used until a better, more effective treatment is discovered.